Whether you believe it or not, hunger and hunger-related diseases are still the main problem. More than 9 million people die every year because of hunger, and that number is much more than tuberculosis, HIV and malaria combined together.
The major factor that limits the food availability are increasing population, not enough land and climate changes.
So that means we have to increase our food productivity. There are different crops which are used worldwide. The most popular ones are maize, sugarcane, followed by rice.
Why we have chosen rice is because it is a staple crop for 1 billion of world's population. It is not just a plate filler, it is also a livelihood for 1/5 of world's population that depends on rice production for their income.
If we stop growing rice today, believe me that we will go to bed hungry. India is the biggest exporter of rice all over the world. 40% of global rice trade is done by India itself. Although it's the biggest exporter, it's not producing rice to its maximum potential. So total yield per plant can be decided by the number of seeds per plant and also by the size of seeds in that plant. So this is the aspect where my lab is focusing more, that is the seed size. In order to increase the size of rice, we have to understand the mechanism, how it is controlled. Cell division, cell proliferation and cell expansion in different dimensions decides the overall seed size. There is a transcriptional cofactor, mediator complex. One of the subunits in that cofactor, MET15, regulates many of the genes which are involved in these processes. And so if you modulate the expression of MET15, you can modulate the size of rice.
We are trying to analyze the epigenome modifications in 2 contrasting genotypes of rice that have varying seed size. Epigenetics is a level of control that sits above the genes and decides which gene is to be turned on and which genes are to be turned off. Conventional breeding over the last several centuries have limited genetic diversity. Epigenome could be one of the major sources of diversity in rice.
We are taking 2 different approaches. One is directly in the field, and that is that we are trying to introduce long grain allele of MET15 into the short grain varieties from eastern part of India. And the other approach, we are doing it in the laboratory, where we are using CRISPR-Cas9 gene editing tool to increase the expression of MET15. The most popular basmati rice in India is Pusa Basmati 1. The grain length of Pusa Basmati 1 is almost about say 7 millimeters to 10 millimeters. Pusa Basmati, it's grown only in the northern part of India and in Pakistan. You cannot grow this in other part of India like southern India or in the eastern part of India. The yield will go down and you will not get the same size of seeds.
The small grain one is Sonasal, which grows in northeastern part of India. It is 3 to 4 millimeter. So we are trying to increase the size of the small grain rice up to the size of long grain rice.
If we introduce MET15 from Pusa Basmati 1 into that eastern variety, the size of that rice seed should increase to say up to 7 millimeters. We may get around 25 to 30% increase in the yield because of just increasing the size of the length of that eastern variety. This will also give commercial benefit to the farmers who are growing this variety. We will be able to increase the income of farmers growing these rice varieties, and also we will be able to feed more people. ICGEB is an international network of over 60 countries, including 3 main components: Italy, India and South Africa.
Here we emphasize at research, the outcome of which helps the society at larger scale.
In this project, we are trying to increase crop productivity, and that's why it is in line with SDG number 2 of United Nations, which is zero hunger. The success in the lab is dependent on the publications and enhancing the knowledge, but the major success can be measured only when that enhancement in the knowledge is useful to the people living in the society.
As we see that the world is facing food insecurity, malnutrition, hunger, we would be able to contribute to a larger population with lesser use of natural resources.